Hi everyone, we will continue exploring the topic Bioinformatics. In this session, we will learn a bit about how protein works and how to visualize protein binding. This presentation is based largely on the information provided on the book Deep Learning for Life Sciences and the example from the DeepChem community. In session 6, we explored several protein visualization options. For instance, we could visualize the protein in default cartoon view. We could visualize the ball and stick model with different amino acid residues colored differently. We could also add on surface view to specific acidic regions on the ball and stick model. In session 7, we will discuss how a protein works in a cell after a small ligand binds to the protein. Proteins play a crucial role in cell signaling, a process essential for cellular communication and function. Cell signaling begins with reception, where a signaling molecule, known as a ligand, binds to a protein, forming a ligand-protein complex. The cell detects the ligand from its external environment, and the ligand then binds to a receptor protein, either on the cell surface or inside the cell. Once the ligand binds to the receptor, it induces a structural change in the protein, triggering signal transduction. This process involves a cascade of molecular interaction where each relay molecule modifies the next in the pathway. Eventually, the signal reaches its target, eliciting a specific cellular response. In essence, an external signaling molecule leads to an internal cellular response by binding to a protein. The intricate three-dimensional structure of proteins enables them to selectively bind to highly specific ligands. This specificity allows ligand to regulate protein function, leading to either positive or negative cellular responses. Thus, understanding the mechanism of cell signaling is a fundamental to drug discovery and development, as targeting these interactions can help design therapies that modulate protein functions effectively. As mentioned, proteins frequently bind to small molecules, a fundamental process that influences their function. For many proteins, their primary role involves binding to specific molecules. For instance, signal transduction in cells often relies on protein binding to other molecules to relay messages. Additionally, when a foreign molecule such as toxin binds to a protein, it can interfere with the protein's function, potentially leading to harmful effect. Understanding how, where, and when molecules bind to proteins is essential for deciphering protein function and developing new drugs. Protein binding involves highly specific interactions, making it computationally challenging to predict. Even a tiny shift in the position of just a few atoms can prevent a molecule from binding. Furthermore, a molecule may only bind in certain conformations, while others may be incompatible. Binding in turn also induces further structural changes in the protein and thus its function. Let's walk through a code example to visualize a protein, a ligand, and their complex using the core dataset from the protein data bank. pdb-bind_core_df.csv.gz. This dataset has been pre-processed and is provided as a gzip file. To begin, we must import the necessary modules: DeepChem for Chem Informatics and Molecular Machine Learning, Download URL module for fetching external resources, Load_from_disk for loading pre-processed datasets, and OS module for handling file operations. Next, we will download the PDB bind dataset from the given URL. First, we retrieve the current working directory using the get_data_dir function from the dc.utils module. Then we construct the full file path by combining this directory with the file name using the join function from the os.path module. Then use the print command. We display the full file path, data_file. The full file path is shown in here. In the next segment, we use an if statement to check whether the file exists in the file path. If the file does not exist in the file path, then it will print file does not exist, downloading file. Then we download the file from the specified URL using download_URL function. This is the source where we download the file, pdb-bind_core_df.csv.gz file. Then we print file downloaded. Now let's inspect the data using the print function. First, we will print the text, type of data is, followed by the data type, which we obtain using the type function. The %s format specifier is used to insert the string representation of the data type. The result is shown here. Type of the data is pandas.core.frame.dataframe class. Next, we print a subset of the dataset, specifically the first entry, index 0. There is a colon and 1. This means that from 0 to 1. By Python's syntax rule, when we specify a range from 0 to 1, the last value 1 is excluded. In other words, we're printing only the first protein-ligand complex entry from the dataset. In other words, this is one row of the data. One row, 7 fields. Next, we print the text, shape of the data is, followed by the dataset shape using the %s format specifier for string data. The %s symbol is used to insert raw_dataset, the shape, which represents the dimension of the dataset. The result is shown here. Shape of the data is 193,7. This means the dataset contains 193 pre-processed protein-ligand complexes, each with 7 features or type of information, 7 columns. To better understand the dataset, let's take a closer look at its structure. Each row represents a molecular system, specifically a protein-ligand complex, while each column provides different pieces of information about that system. For example, PDB-ID represents the protein data bank identifier. complex ID represents a unique complex identifier. Smiles represent the smile string of the ligand. Label represents the binding affinity of the ligand to the protein. The dataset contains the following column headings: PDB ID, smiles, complex underscore ID, protein underscore PDB, ligand underscore PDB, ligand underscore MOS2, and label. To visualize the protein ligand and their complex, we need to import a few additional modules. Angle View module for interactive molecular visualization. Temp file module for handling temporary files. OS module for file system operations. MD-TRAS for working with molecular dynamic trajectories. NumPy for numerical computations. Pandas for data manipulation. We will define a function here. Convert underscore lines underscore to underscore MD-TRAS. This function extracts and processes information from a specific field within the PDB bind dataset, converting it into a format compatible with MD-TRAS for molecular analysis. Let's take a closer look at how the convert underscore lines underscore to underscore MD-TRAS function works. The function first processes the input by removing the square brackets, single quotation marks, new line characters, and splitting the cleaned data using a comma as a separator. Then it creates a temporary file. It generates a temporary directory using mkd temp function from the temp file module. Then it combines this temporary path with the file name molecule.pdb to construct the full file path stored as a molecule underscore file. The function then opens the file at the specified full file path using the open function in write mode, allowing us to write the processed molecular data into it. Also, the file is referenced as the letter F. Next, we use a for loop to write each line from molecule underscore lines into the temporarily opened file. Now the data written to the molecule underscore file is loaded into an object called molecule underscore MD-TRAS, which is returned from the function convert underscore lines underscore to underscore MD-TRAS. Now let's see how we use our newly defined parsing function convert underscore lines underscore to underscore MD-TRAS in action. First, we extract specific information from raw underscore dataset and store it into first underscore protein and first underscore ligand. We use this ilog function with index 0 to select the first entry, first row information in the raw dataset. And then this is specifying the field protein underscore PDB or ligand underscore PDB. So in summary, we are selecting the first row and then protein underscore PDB column, first row and ligand underscore PDB column, and store it into this variable first underscore protein, first underscore ligand. Next, we apply our convert underscore lines underscore to underscore MD-TRAS function to parse this extracted data. This prepares the first protein and the first ligand for visualization using angle view and MD MD-TRAS. The prepared information is stored in the protein_MD-TRAS, ligand_MD-TRAS. Now we can visualize the three-dimensional structure of the ligand using the show_MD-TRAS function from the angle view module. Similarly, using the same function, we can also generate a three-dimensional view of the complex protein structure. Next, we need to visualize the protein-ligand complex. Just like we created a function for parsing, we now define a function for this visualization process. This will allow us to reuse it efficiently throughout the code for different protein-ligand complex. Here we define a function called combine_MD-TRAS, which adds a ligand to a protein to construct a protein-ligand complex. The descriptor object chain adds a chain to the protein topology using the addChain function from the topology class. Next, the residue descriptor adds a residue to protein topology with the add_residue function, also under the topology class. This add_residue function takes three arguments: name, chain, and residue sequence. Name represents the name of the new residue, which is set to LIG, representing the ligand. Chain represents the protein chain to which the ligand residue will be added. Res_seq represents the residue sequence number, which defines the order of the residues in the chain. In this case, the sequence number is 1. Once we have created the necessary descriptor instance objects, the function uses a looping structure to iterate through all the atoms in the ligand molecule's topology. It then adds these atoms to the protein topology as part of the same residue we previously defined. This step effectively connects the ligand to the protein, creating a new protein-ligand complex topology. The actual atomic coordinates for these atoms will be determined in the later steps. Next, we horizontally stack the Cartesian coordinates of the atoms in the protein and ligand objects and assign the result to protein.xyz. The xyz attribute of the atom group object in MD analysis holds an array of shape, n atoms, and xyz coordinates, so 3 fields. So n, 3, which contains the coordinates of all the atoms in the group. To combine these coordinate arrays, we use the hstack function from the NumPy, which horizontally stacks the arrays along the second axis. Essentially, it appends the columns of one array to the right of the another. In this case, both protein.xyz and ligand.xyz have the shape n number of atoms, 3 field xyz, 3 coordinates. So when we horizontally stack them, the result will be an array with the shape n number of atoms and 6 fields, xyz for protein and xyz for ligand. By assigning this concatenated array to protein.xyz, we combine the Cartesian coordinates of both protein and the ligand into a single array. This array now represents the coordinates of the entire protein-ligand complex. Next, the create_standard_bonds function found under the topology class uses a simple algorithm based on covalent ready to establish standard covalent bonds between atoms that are within a certain distance of each other. As a result, the protein topology object is updated with the bond formation, reflecting the covalent radii of its constituent atoms. So what the function is returning is the protein that includes the ligand. In the next line, we use combine_MD_TRAS function to create the protein-ligand complex. Finally, we can visualize the protein-ligand complex using the AngelView.show_MD_TRAS function. As you can see that the ligand has found its position in the protein cavity. Okay, let's run the same code in the Jupyter Notebook platform. We first import the necessary module. We download the PDB dataset from a specified URL. We load the data into a data frame called raw_dataset. We inspect the data using the print function. We define a parsing function for the PDB dataset. Then we visualize specific ligand and protein. Then we define another function combining the protein and ligand. Then we visualize the protein-ligand complex. Let's run the same code by running all the cells. As anticipated, the ligand is visualized, protein is visualized, protein-ligand complex is visualized. I can zoom in to see the ligand-protein binding. Okay, it binds to the back side here. So if I rotate it in this direction, you can see that the ligand right here. Okay, this is the first protein-ligand dataset. You can change it to the specific protein and specific ligand by changing the index number here. Let's change it to 11th protein and 11th ligand. And we can run it from this cell below by clicking on the Run Selected Cell and all below. This is the 11th ligand and this is the 11th protein. And this is the 11th protein-ligand complex. You can see the small ligand inside this cavity of the protein. Let's zoom in and see. The ligand is right here. It binds to the cavity of the protein. This is the end of the session. We learned how protein works by binding to a ligand, and we visualized protein, ligand, and protein-ligand complex. Thank you for your attention. I hope you found the session engaging and informative.